Distinct E-cadherin-based complexes regulate cell behavior through microRNA processing or circ and P120 cantonine activity, or restoring proper function of cancer cells. So special thanks to Luke D., Ms. Legerfo, Sydney Stein, Mark Strockman, the Journal Club team, my mom, and my family. So first, what is cancer? Can anyone name some specific characteristics of cancer? Interactive. So there are 5 main characteristics of cancer cells, one being accelerated proliferation. So cancer cells have the ability to divide and grow uncontrollably, and that is accelerated proliferation. Angiogenesis is the recruitment of blood vessels. The reason that cancer cells do this is to support this division and growth by feeding itself with those blood vessels. The next is the resisting apoptosis. So when a cell is growing and dividing, it goes through many checkpoints to make sure that there's no mutations in the DNA, it's going to be a properly functioning cell, etc., etc. But these cancer cells find a way to go around those checkpoints and don't commit suicide, which is what apoptosis is, cell suicide. So they don't commit suicide like a normal cell would when it's not functioning properly. The next 2 are very important to this paper, both having to do with motility or movement of the cell's mobility. So number 1 is invasion. So if we're talking about melanoma or skin cancer, invasion is when it starts on the top layer and goes through multiple layers of skin. So that's invasion. Metastasis is almost the next level of invasion, so it goes through those layers and then into the bloodstream. But the thing that makes it metastasis is when it starts a new tumor somewhere else. So a melanoma tumor could then become a brain tumor when it travels through the bloodstream to the brain and then starts its own colony. So those are the 5 characteristics. So I want you to remember to keep those 2 in mind that have to do with motility. So the current therapies are surgery, chemotherapy, and radiation. But what do they all have in common? Interactive. So all these current cancer treatments focus on killing cells. They focus on killing cancer cells, but the problem is they can't target those cancer cells specifically. So they're highly toxic because they don't always only kill those cancer cells, they also kill the regular cells, which is why they become invasive and intrusive, because you have these extreme side effects of these treatments. But also your body can become resistant just like any other treatment, because a treatment that once worked often stops working, which makes it resistant. Now diving into the paper, I want you, from those treatments, I want you to remember that those treatments are only focused on killing the cells, but they're problematic because they're highly toxic. But now we're going to dive into the paper and talking about E-cadherins. So this paper has to do with epithelial cells. So epithelial cells are formed in layers. So E-cadherins are epithelial, E-cadherins adhere. So these help the cells adhere to each other and stick together. So when these are present, when these E-cadherins are present, they stop the cells from becoming motile, as we talked about earlier. They cannot move, which are 2 very basic characteristics of cancer. They can't move when these E-cadherins are present. The problem is P120 stabilizes these adherins. So with P120, they're locked together, and without P120, they can be absorbed into the cell. Therefore, without these adherins, that's when they are able to become motile. So yes, with E-cadherin and P120, it's a tumor suppressor. But when P120 is present, it's also been known to have oncogenic abilities, which means it can promote tumors. So it's very contradictory and we don't know why. So the original purpose of this paper is to look into that. So what they start by doing is they know that pleca is only found apically, so they decided to localize these proteins. So pleca, as you see in green, is only found apically, but in red, P120 is found apically and basolaterally. So apically is the top, basolateral is the sides and the bottom. And the figure on the right also shows this. A silver stain is very similar to a western blot. So the green is apically, the red is basolaterally. So pleca only has green, therefore it's only found apically. P120 has both red and green, therefore it is found basolaterally and apically. So we're going to start to build a pathway here. So P120, it's found apically and basolaterally. So basolaterally, we know that the purpose of P120 is to stabilize that cell-to-cell adhesion. But what does it have to do apically? So that's what we want to look into. So we look into pleca. So we knock down pleca to see what effects it has on the cell. So on the right here, you can see stained cells. So on the right actin, it has to do with the cell structure. Pleca on the left, it's stained. So on the bottom, pleca shRNA is when it's knocked down. So when NT is the control. So what do you notice the difference between the knockdown of pleca and the control of pleca? Just looking at the figure on the right. Yes, so the structure is messed up and without pleca. So the structure doesn't have its original integrity. What affects, so if you look on the y-axis of C here, fold change just means the proliferation and the cell growth and division. And then the first column is the control, while the other two are the pleca knockdowns. What does this tell you? Yeah, so this is an interactive slide. So yeah, when pleca is downregulated, there is an increase in cell growth and cell division, which leads to tumors, because that's one of the characteristics of cancer, accelerated proliferation. So we're going to go back to that pathway. So, okay, pleca somehow has to do with stability, structure, cell growth, and colony formation. So when pleca is downregulated, the structure and stability are decreased, while the cell growth and colony formation are increased. Bad things, not happy, Kanye. So we wanted to look into why that is. So next we did more knockdowns. So what happens when they knocked down pleca and P120? So here you see the negative signs. So pleca, so shRNA have to do with knocking them down. So when it's pleca shRNA, it's knocking down pleca. P120 shRNA, knocking down P120. So when it's negative, that means they're not using those knockdowns. But when they're positive, it means that the knockdowns are being used. So based on A, what does this tell you? If fold change means the same thing, proliferation, cell division. Interactive. Yeah, so when it's control and nothing is being changed or deregulated, there's normal cell growth and division. When pleca, as we saw earlier, is downregulated only, there's an increase in cell proliferation. But when there's a double knockdown of pleca and P120, the normal cell growth and division levels are restored. Why is that? So now we look into B. So B is talking about what's boxed in blue is these growth factors. So same as on the left, on the bottom, it's the double negative is when it's not being used, when the positive, the knockdowns are being used. So what do you see here? What happens when both pleca and P120 are knocked down with the growth factors? Interactive. Yeah, so P120, and when pleca and P120 are both knocked down, the levels of those growth factors are decreased, which would make sense when comparing it to A, because if the growth is decreased, then it would make sense that the growth factors are therefore decreased as well. So now we're going to add that back to the pathway. So P120, apically, so pleca and P120 working together, both upregulated, or both normally regulated or both downregulated, you can see that the growth factors are properly expressed, and so pleca and P120 somehow have to do with inhibiting these growth factors. That's what the upside down T means. So now we want to look up why it's being downregulated. So what does that mean? So we knew that pleca had something to do with microRNA. So microRNA they also regulate proteins and the expression of these proteins. So they looked at, okay, when pleca is knocked down, what microRNAs are also knocked down or also increased? So what are the change in RNAs when pleca is also changed? So we noticed that the most dramatic of all of the microRNAs was the microRNA 30B. It was significantly decreased when pleca was knocked down. So we wanted to see, okay, what happens to these growth factors when we can mimic the expression of those microRNA 30Bs? So we have on the left wild type, we have the second column, we have a normal expression of pleca with they mimic microRNA 30B added, and you can see they're pretty much similar. So, and then with the pleca knockdown, we have a control without the mimic microRNA, and then with the mimic microRNA, excuse me. So what you see here is that without the microRNA, as we saw earlier, these growth factors are increased dramatically. But when you compare it to, but then when you add the mimic microRNA 30B, it restores the growth factors to their normal levels. So that means that it's with the insertion of microRNA 30B, we have now essentially restored the properly growing cells. So now we're going to finalize this pathway, and this was actually found in the paper. So this proves what we're saying here, that pleca and P120 and E-cadherin lead to microRNA 30B, and microRNA 30B inhibit, that's what the upside down T means again, they inhibit those growth factors. So what it's also saying is that pleca represses P120 oncogenic behavior, or the expression of pleca suppresses the P120 to act as a tumor promoter. So what does all of this mean? So we have now clarified the roles that P120, E-cadherin, and pleca have in cancer. And the big thing here is that we have now, by mimicking these microRNA, we're able to rehabilitate these cancer cells. So essentially, we have made these cancer cells revert back to acting as normal proliferating cells. So what's next? This is a potential therapy that needs to be tested in vivo. But the big thing here is we can find additional, less aggressive treatments, because now we don't have to treat and kill all of these cancer cells, but what if we can revert these cells back into normal cells? We have essentially found our first example of being able to reverse cancer, and that's a huge thing that we still need to look into. Questions?